Welcome back to another episode of Cancer as a Mitochondrial Metabolic Disease Basics. Most people, even healthcare professionals, think of melatonin as a sleep hormone. Take it at night, maybe sleep better, maybe improve your circadian rhythm. But as we've seen in prior videos, melatonin is doing something much more interesting in cancer biology. However, in this video, I want to show you something most people have never seen. There are studies showing that melatonin can reduce cancer cell glucose and glutamine uptake, suppress glutamine metabolism by lowering glutaminase expression, and interfere with the two major fuels cancer cells depend on, glucose and glutamine. But there's one question that almost nobody asks. What concentrations did they use? Because if a study uses 0.01 or zero point 0.1 or 1 millimolar of melatonin in a petri dish, that does not automatically mean a person taking 3 milligrams before bed is going to get anywhere close to that in the bloodstream. So in this video, we're not going to just ask, can melatonin affect cancer metabolism? That appears to be an obvious yes. We're going to ask the harder question, can oral melatonin realistically reach the concentrations needed to inhibit glucose and glutamine metabolism in cancer cells? So without further ado, let's get into it. So as I alluded to in the prior video, there's just too much to share about melatonin and what it can do for us, especially in the context of cancer, to put it in one video. And although the last video was pretty eye-opening about some of the molecular mechanisms of how melatonin can help us, specifically in the context of cancer, looking at through a metabolic disease lens, I think this video may even be more enlightening for us. So I'm going to start out with a paper that was published in November of 2024. It's titled SLC38A5 suppresses ferroptosis through glutamine-mediated activation of the PI3K AKT mTOR pathway, signaling in osteosarcoma. And it says here that SLC38A5 is upregulated in osteosarcoma and is associated with poor prognosis in patients. Upregulation of SLC38A5 promotes proliferation, migration, and invasion of osteosarcoma cells, while the PI3K inhibitor BKM120 can counteract these effects. Additionally, silencing of the SLC38A5 inhibits tumor growth in vivo. So I want to get us a bit oriented here. We have in the prior videos, and this is going back a ways because we're talking glutamine pathways, but we talked mostly about a transporter called SLC1A5. And most of the things that we have got at our disposal all affected SLC1A5. And we saw it more recently when talking about oxygen at decreasing SLC1A5 expression and therefore decreasing glutamine uptake into cancer cells. But what we have not talked about is SLC38A5. This is something that I've never seen anything that we have the ability to either ingest or do or take to affect this particular transporter. So this is a novel kind of like mechanism of how glutamine is lowered into cancer cells. Now, there was a paper that was published more recently in 2026. Actually, it was very recently published in 5/11/2026, so just published a few days ago. And it was titled Impaired Redox Adaptation through SLC38A5 Dependent Glutamine Metabolism Contributes to Melatonin-Mediated Oncolysis Sensitivity and Metastasis Suppression in Bone Cancer. And what it said here was, in the abstract, malignant bone cancer, primarily osteosarcoma and chondrosarcoma, are highly aggressive neoplasms originating from bone tissue and characterized by a strong propensity for lung metastasis. Tumor cells evade anoecus through reactive oxygen species-mediated redox homeostasis, which modulates signaling cascades that promote proliferation, survival, and metastatic invasion into distant sites. Melatonin, primarily synthesized by the pineal gland, that's kind of old news, and has been implicated in cancer prevention and therapy due to its inhibitory effects on bone cancer growth and progression. Little is known about the mechanisms underlying resistance to this anoecus in bone cancer cells or whether melatonin can therapeutically modulate this process. We demonstrated that glutamine metabolism is essential for bone cancer cells to maintain an weakest resistance. Melatonin treatment disrupted glutamine metabolism and altered redox homeostasis, as evidenced by increased ROS accumulation and reduced NADPH NADP ratios under anchorage independent conditions. Notably, solute carrier 38 member 5, SLC38A5, a glutamine transporter, was identified as a critical regulator of bone cancer progression, with higher SLC385 expression correlating with poorer clinical outcomes. Melatonin suppressed SLC38A5 expression and attenuated anoecus resistance through inhibition of the PI3K AKT signaling pathway. Consistently, reduced SLC38A5 expression was associated with decreased lung metastases in melatonin-treated groups in an orthotopic mouse model. Collectively, our findings revealed that a previously unrecognized role of melatonin in modulating glutamine-dependent redox balance and anoecus resistance in bone cancer. This study highlights SLC38A5 mediated glutamine metabolism as a critical determinant of metastatic potential and supports melatonin and SLC38A5 as a promising therapeutic target for osteosarcoma and chondrosarcoma. So this is the graphic that was associated with the paper, and it's basically showing anywhere where melatonin is at here, and you see like a little blocking sign, melatonin is blocking those particular processes. But I want to refocus back up on this area here, where melatonin, through an inhibition of the PI3K AKT mTOR pathway, there's decreased expression of this glutamine transporter SLC38A5, and therefore there is less glutamine getting into the cancer cells because of that mechanism. And this would be a novel, not understood mechanism prior to this paper coming out, which I thought was pretty cool. So let's now break down what they did in these experiments. So the first thing they looked at was a combination of different types of osteosarcoma cells, this JJ012 and this 143B types of cells. And we see that they gave varying concentrations of melatonin in millimoles. Okay, so there's a 0.1, 0.3, and a 1 millimole of melatonin used. And then there are drops in glutamine concentrations inside the cells when they give varying degrees of melatonin. So basically at 0.1 millimole, there was a 15% drop in glutamine uptake. At 0.3 millimoles, there was 23%. And at 1 millimoles, there was 37% decreased glutamine uptake in this cell line of osteosarcoma or bone cancer cells. Let's look at this 143B. So if they gave 0.1, it dropped by 12% the glutamine uptake. At 0.3 millimoles, it dropped by 18%. And at 1 millimole, it dropped 40% from baseline when given melatonin at that concentration. This is another mechanism that I would have imagined would have been possible, given we know melatonin's effects on MYC and HIF and AKT and all these other signaling pathways. But this is actually a study showing that melatonin in both cell lines decreased glutaminase, the enzyme that DAWN directly blocks, by 33% and 45% respectively in either this JJ012 or the 143B osteosarcoma line. That's a significant amount. We're not talking about just decreasing glutamine uptake by cancer cells. Now we're talking about the necessary machinery to convert that glutamine into glutamate so it can be used by the cell to either make energy through fermentation or to help support redox homeostasis. Pretty unbelievable. Let's take a look here at some other studies they did. So here is the amount decrease of SLC38A5 depending on whether melatonin was added or not. So if it didn't get melatonin, let's say it was 1, and at 1 millimole of melatonin, it dropped by 30 to 35%. And then for this other cell line, the 143B, at 1 millimole, it dropped 45 to 50% the expression of this transporter. Okay, so this part of the study is not looking at percent glutamine getting into the cell. It's now saying what percent of this transporter is now decreased when we give melatonin. And that's what they're looking at here. It's 30 to 35% for 1 millimole and 45 to 50% decrease at 1 millimole for this other cell line. Now, what's the other thing they did? They looked at specifically at 1 millimole how much decrease in glutamine uptake was done under melatonin influence. And again, it was 30 to 32% in this particular experiment, and it was 38 to 40% in this cell line. Again, this is at 1 millimole. Keep that in mind, 1 millimole. So the next paper I wanted to pull was published in August of 2025. It's titled Melatonin-Induced Ferroptosis in Pancreatic Cells by Simulating Endoplasmic Reticulum Stress and Inhibiting Alanine, Serine, Cysteine Transport 2-Driven Glutamine Metabolism. And it says here, additionally, in the abstract, melatonin activated the endoplasmic reticulum ER stress pathway, inhibited glutamine metabolism, and promoted ferroptosis in pancreatic cancer cells. And how did it do that? Well, I kind of used the paper itself to help us answer that. So after 24 hours of exposure to melatonin, the amount of SLC38A5, the other more classical glutamine transporter, was decreased. Glutaminase and XC, or SLC3811, also known as the Achilles heel of cancer, was also decreased, as well as glutathione synthetase and glutathione peroxidase. So variety of ways that melatonin is inhibiting the glutamine pathways that help balance the ability for the cancer cells to maintain redox homeostasis, the balance between reactive oxygen and reactive nitrogen species and antioxidant systems to help them stay alive, right? So melatonin is collapsing that. And if I took it on a graphic, so we see that melatonin is acting on SLC1A5, forgot to put an arrow there. It's working on this SLC38A5 as well as SLC7011, the Achilles heel of the tumor. And then it's also having an effect on glutaminase, as well as other enzyme systems in the glutathione pathways that are not even on this graphic. Astounding. Now let's switch gears to glucose. This paper was published in July of 2017. It's titled Melatonin Decreases Glucose Metabolism in Prostate Cancer Cells, a C13 stable isotope resolved metabolic study. And it says the pineal neuroindole melatonin exerts an exceptional variety of systemic functions, some of which are exerted through its specific membrane receptors, type 1 and type 2 melatonin receptors, while others are mediated by receptor-independent mechanisms. This is important. There's receptor-mediated and receptor-independent non-mediated effects of both melatonin and of vitamin D as well. A potential transport through the facilitated glucose transporters GLUT SLC2A1 was proposed in prostate cancer cells. The prostate cells have a particular metabolism that changes during tumor progression. Actually, all cancer cells do this. During the first steps of carcinogenesis, oxidative phosphorylation is reactivated, while the switch to the Warburg effect only occurs in advanced tumors in the metastatic stage. That's to be debated. Here, we investigated whether melatonin might change prostate cancer cell metabolism. To do so, we used 13-carbon stable isotope resolved metabolomics in antigen-sensitive LNCAP, which is a specific type of metastatic prostate cancer. I'll define those in a minute. And insensitive PC3 prostate cancer cells were employed. In addition to metabolite C13 labeling, ATP, AMP, and lactate dehydrogenase or pentose phosphate pathway activity were also measured. Melatonin reduces lactate labeling in androgen-sensitive cells and lowers C13 labeling in TCA cycle metabolites and ATP production. In addition, melatonin reduces lactate, C13 labeling in androgen-sensitive prostate cancer cells. Results demonstrated that melatonin limits glycolysis as well as the TCA cycle and pentose phosphate pathway in prostate cancer cells, suggesting that reduction of glucose uptake is a major target of the indole in this tumor type and every other tumor type. So let's take a look at what they found. So they had two separate prostate cancer models. One was this LNCAP, which is a metastatic prostate cancer to lymph nodes, and metastatic prostate to bone was PC3. And I believe that PC3 is also castrate resistant or non-androgen receptor sensitive or active. So that kind of differentiates the two models we have here. And we'll see here that at, again, varying concentrations of melatonin. Here's the control, 0 melatonin. Here's 0.01. Here's 0.1. And here's 1 millimole of melatonin. we see decreases in glucose uptake by this lymph node metastasis prostate cancer model and this bone metastasis prostate cancer model. And we see here that in A, at 0.01, there's a 27% decrease in glucose uptake. At 0.1, there's a 27% decrease uptake in glucose. And at 1 millimole, it's an astounding 79% decreased uptake of glucose by melatonin alone. Let's look at the metastatic prostate cancer that's bone metastasis and androgen insensitive. You see that at 0.1, you get a 25%, you get about a 23%, and at 1 millimole, you get a 37% decrease in glucose uptake by these cancer cells. I'm going to ignore this side of the graphic because basically what they did is they took melatonin alone, and then what they did is they added glucose to it. And of course, there's going to be more glucose being uptake when there's more glucose available. That's what HG stands for, high glucose. And what they were basically showing was that if you gave high glucose and melatonin, you still saw a massive effect in both models. Next paper, published in August of 2015, melatonin cytotoxicity is associated with Warburg effect inhibition in Ewing sarcoma. And I'm just going to skip to the graphic here in this particular paper. So we see here that in this T71, this is what A represents, T71 Ewing sarcoma. There's 3 different cell models they use in this. I had to kind of like tease through this. We saw that if melatonin was given, and I believe they use 1 millimole, I'll have to look at it again. They got a 32% reduction in glucose uptake by this type of Ewing sarcoma cells. Then they looked at the other 2 cell types, and at 8 hours, the A4573 Ewing sarcoma had a 23% reduction in glucose, and the A673 had a 19% reduction in glucose by the same concentration of melatonin at the same amount of hours, 8 hours, which I thought was pretty astounding. And this was for a different type of cancer altogether. So this led me down rabbit holes to try to figure out what amount of dose matters to try to get those concentrations used in those studies. So I use AI to help. Like it or love it, it helps me with this math. So it's not perfect, but it's a heck of a lot better than I would be doing if it was me with a calculator. So I plugged in what would be the amount of immediate release versus liposomal or micellar melatonin to achieve 0.01 millimoles. And to get 0.01 millimoles, you would likely need 2.5 to 5,000 milligrams of immediate release and 1 to 2,000 milligrams or 1 to 2 grams of liposomal. To get 0.1, you need 25 to 50 grams or 25 to 50,000 milligrams to get 0.1. And to get 1, which is kind of the standard, what they use in a lot of these papers, to get 1, you need 250 to 500 grams. If you're using liposomal, you need 80 to 200 grams. This is basically impossible. This is damn near impossible too, let's just be honest. And this is possible, but it's just like very unlikely that we're going to take 5,000 milligrams of melatonin per day. Let's look at what Gemini said. It basically came up with the exact same math. Normally AI models don't come up with the same math unless there's just a lot of like literature and the pharmacokinetics are fairly worked out with what is possible for a specific compound or drug or supplement. And it seems like melatonin has pretty good data for pharmacokinetics. So it was able to actually figure out that it's about the same. Let me tell you why I think that doesn't matter. Okay. Now this is the downside. This is the upside. Okay. It is known that whatever we put into the bloodstream, about 100 times more gets into the cell and specifically inside the mitochondria. Why does that matter? Because if we were to make that calculation, if what would be the oral estimated dose needed to get the concentration we want inside the mitochondria, where it actually matters anyway, it tells a very interesting story. To get 0.01 millimole, we would need 5 to 10 milligrams of normal melatonin and about 3 to 5 milligrams of liposomal. To get the 0.1 millimole, we would need 250 to 500 milligrams of normal melatonin and 100 to 250 milligrams of liposomal. And to get the 1 millimole, the gold standard kind of concentration they use to get the most dramatic effects, of melatonin on glucose and glutamine, then you'd need about 2,500 to 5,000 milligrams of melatonin, normal melatonin. And to get the same effect, you'd probably need somewhere about 1,000 to 2,000 milligrams of liposomal. And basically, the numbers ended up being nearly the same for Gemini and ChatGPT. So, what does that mean? Let's go back to the actual studies. Let's use this one instead. Okay. So, I think for most people, 1 millimole concentration of melatonin is very unlikely. Even with that 100x multiplier of how it aggregates within mitochondria specifically, the 100x multiplier, I think the 1 millimole is probably out. I do think that zero point zero 0.01 and 1 are very within our reach. And that still gives us significant decreases in glucose uptake, right? Here's 27% and 27% respectively. If we go down to the glutamine studies, we saw that at zero point 0.1, that gives us about a 15% and a 12% decrease glutamine uptake within these osteosarcoma cells. So my point by showing all of this is that let's say most people probably could get the 0.1 millimoles. That would take a 250 to 500 milligram estimated dose, and the half-life is not super long with this, so it may require repeated dosing, which means that people who are doing the 1,000 milligrams per day or whatever, or multiple grams per day, are probably getting above the 0.1, probably never going to get up here to this 1 millimole. I mean, maybe if they're doing like multiple thousands per day, but they're still getting to this 0.1 millimole probably. And that is probably true, right? And you're probably getting those effects that you're seeing there. You're just not getting these 1 millimole concentrations that give these very dramatic, effects, the 37%, 40% reduction in glutamine, the 35 and 45% reduction in glutaminase enzymes, and the other graphic showing like, 80% reduction in glucose. That probably is not realistic, to be very honest with you. But these 0.1, I think, is well within range. It's just that it's going to require a supplement or an intervention, which I'll talk about when we talk about boosting our endogenous melatonin. I have some interesting data around that as well. But I think it's going to require probably, they make these like 120 milligram capsules. They may even make higher ones. Those are the ones I'm aware of. But it's going to require those because otherwise you're taking like 3 milligram capsules, like 100 of them or something like that. Like no one wants to do that. But the 120s make it more accessible to get these kind of numbers. And anyway, so that's what I got. I hope this was helpful, and I hope it made it more realistic about what kind of results we can expect at lowering glucose, at lowering glutamine levels with melatonin, with kind of realistic oral doses. And anyway, if you like videos like this, please like, share, and subscribe. And until next time.